you have to bear with me a little because I'm doing this with my other class as well, and we're at different places. So, did we talk about polysaccharides already?
Yeah.
We did. Okay. Any questions on carbohydrates then? Okay, so remember carbohydrates. There can be a few different forms. Monosaccharides, which are just one carbon chain, one sugar molecule like glucose or deoxyribose. Disaccharides, which were a type of oligosaccharide. Disaccharides have two monosaccharides that are linked together. Di for two. We looked at lactose with that and probably sucrose. And then the polysaccharides, which are many carbohydrates linked together, many monosaccharides, many glucose molecules linked together to make up a polysaccharide like starch or glycogen. The second group of organic molecules are the lipids, at least the one that we'll talk about, right? So lipids are fats. If we were to look at this in terms of the body, the majority of the fatty tissue in the body, called adipose tissue, is composed of lipids, such as the triglycerides. Okay. The cell membrane, the plasma membrane of each of the cells in your body, is composed of lipids. These are phospholipids, and we call this membrane the phospholipid bilayer. Some of the hormones that we'll talk about next semester, like the steroid hormones, estrogen, testosterone, these are composed of lipids. These are the sterols. They have a basic 4-carbon ring structure to them. Okay, so all of these different examples are fats. They are lipids. Lipids are non-polar. So do they like water?
No.
No. They don't like water. They do not dissolve easily in water, and in fact, they will repel away from water. They'll turn away from water so that they are protected or least exposed to the watery environment. We will look at them in terms of energy storage, specifically with the triglycerides. They can store the most energy. We'll also look at them as the building block, the membrane surrounding your cells, and as a coating, a protective coating in different areas of the body. Most areas of the body will have a layer of adipose tissue or a layer of fat for insulation and energy reserve and extra protection around them. Okay, so looking at some examples of lipids, in particular, we'll talk first about phospholipids and then we'll go into the triglycerides. Both of them are built using fatty acid tails. We call these fatty acid tails. They're essentially long chains of carbon atoms that have single bonds in between the carbon atoms and the hydrogen atoms, and in some cases can have double bonds in between some of the carbon atoms. Now, all the bonds that you are looking at here are covalent bonds. Covalent bonds involve the sharing of electrons, and in a single covalent bond, there's the sharing of one pair of electrons. But in a double covalent bond, like you can see here, we are sharing 2 pairs of electrons, or 4 electrons total. Because we have this double covalent bond right here between these 2 carbon atoms, we do not have another hydrogen atom with a single covalent bond off to the side of each of these. Does that make sense? Because remember, the carbon can bond to up to 4 other atoms, and we want to have 4 lines, essentially, coming from that carbon. Because it's double bonded here, we have 1, 2, 3, 4 coming from that carbon. And this one here, 1, 2, 3, 4. So this is stable. Okay? This particular chain of carbon atoms with the double bonds is different in terms of its structure from this one here with all of the single bonds in between the carbon atoms. Specifically because there's more hydrogens there here in this one than there are in this fatty acid tail. we say that this fatty acid, this chain of carbon atoms, is then saturated with hydrogen. Because at every place that there could be a hydrogen, there is one, because there's only these single covalent bonds in between the carbon atoms. So this fatty acid tail, is called a saturated fatty acid tail. Everyone with me on that? Okay, this one here then is, you tell me, unsaturated. It's an unsaturated fatty acid tail. It does not have as many hydrogens here. And the unsaturated fatty acid tail can be mono-unsaturated, or it can be poly-unsaturated, just depending on how many double covalent bonds you see in between the carbon. There's 1, 2, 3, there's 3 of them here, so this is a poly-unsaturated fatty acid tail. You are not going to have to draw these, but if you see them, you should be able to identify which is saturated and which is unsaturated. Always look either for the double bonds or for the amount of hydrogens bonded to the carbon. Now notice in this unsaturated fatty acid tail, at every place that there is a double covalent bond in between the carbon atoms, the tail is not straight. Instead, where there's a double bond, the tail bends. There's a kink in the chain. You guys see that all right? So this one's nice and straight. It's saturated. There's no bends, there's no kinks. But this one, there's a bend this way and another bend over here. At every point where there's a double bond, there's a kink in the chain. And this is going to affect how they sit within your membrane, within your cell membranes. You might see the fatty acid tail straight like this because they're all saturated, or you might see them bending. At the very top, shown in orange, is the portion of the glycerol molecule that this fatty acid tail is attached to. And you'll notice at the top of this, what functional group is here?
Hydroxyl.
Okay, looks like a carboxyl functional group, doesn't it? A C with a double bond to oxygen, and then an OH, carboxyl functional group. Okay. Now take a look at the different forms of lipids. I said we would compare the triglycerides and the phospholipids. At the very bottom, you can see a triglyceride. Tri for 3. And in a triglyceride, we have 3 fatty acid tails attached to this glycerol backbone. That's where the glyceride portion of the name comes from. So we have 3 fatty acid tails attached to our glycerol. Right? You can still see a piece of what was that carboxyl functional group. And now we have actually formed a special type of bond called an ester link in between the glycerol and the fatty acid tail. You don't have to remember the ester link or the location of the ester link that it's between a carbon and an oxygen. But just so that you recognize that this portion that's highlighted in green is that region of glycerol, and that this portion highlighted in gray is the region of the fatty acid tail. And then compare the 3 tails that you see. Are they all saturated? No. This one is saturated, obviously. There's only single bonds here with the hydrogens, and so is this one. But this one is an unsaturated fatty acid tail. That double bond here between the carbon atoms kinks that chain, and so it's bending a little bit away from the other ones.
So whenever there's a kink, it's not going to be unsaturated?
It's going to be unsaturated.
Because there's a double bond on the...
Correct, because there's a double bond. And if you look at this tail here, there is only one double bond in between the carbons. So is this polyunsaturated, monounsaturated? It's a monounsaturated fatty acid.
So is a triglyceride just 3 of the tails together?
Yeah.
It doesn't have to be those
It does not, you're correct, right? It can be any combination. They can be, monounsaturated, saturated, polyunsaturated. There could be any combination there. But there is 3 fatty acid tails attached to glycerol. That's why we call it a triglyceride. Yep, that's good. Right, so this is what you're storing in your adipose tissue. These long chains of carbon atoms. Why are we storing these long chains of carbon atoms? What eventually can we do to fat?
Break it down.
We can break it down if we need to into smaller chunks and use that for ATP. Just like we would take glucose as our energy source with the 6 carbon atoms in glucose and break that up to get energy, we could do a similar process to this. Yeah.
Why is that monounsaturated?
Sure, because there's only one double bond here in between the 2 carbon atoms. If there was more than one double bond, then it would be a polyunsaturated
So
fat.
more than one king.
More than one king, yeah. Now at the very top, right, this looks almost like a phospholipid, where a phospholipid is a different type of lipid that has 2 fatty acid tails attached to glycerol. I'll show you a picture of that. So you can see what I mean. So here's the glycerol region, those 3 carbon atoms, and here's the 2 fatty acid tails attached to that glycerol. This is a phospholipid. If we were to draw it in a more shorthand form, it would look like this, where we have something similar to a head and 2 tails coming from that. These 2 tails are the fatty acids, and just like with the triglyceride, they can be unsaturated or they can be saturated. There can be different combinations of them. Both of them can be saturated, both can be unsaturated, and we can have a mixture like you see here. Just like with the triglycerides, whenever there is an unsaturated fatty acid, there is a bend or a kink at the point of the double bond. Now look more closely at the head region, where glycerol is. Where's glycerol? There's another functional group over here. And this is one of the ones that you needed to know. Okay, this is the phosphate group. Phosphate group's part of the head of the phospholipid with some other, atoms in there as well. The head region of a phospholipid is different in terms of its activity than the tail regions. The tail regions are true lipids. They're nonpolar. They don't like water. But the head region is actually hydrophilic. It's going to like water. So when we have an arrangement of these phospholipids in the cell membrane, in the plasma membrane, the head regions will turn towards the watery environment on the outside of your cells, and they will also turn, and you can see that here, towards the inside of the cell contents, which is the cytoplasm or cytosol, the fluid interior of the cell. And we have 2 layers of these phospholipids arranged tail to tail. Does that make sense? The tails of one layer of phospholipids are pointing towards the tails of the other layer. On one side of the cell membrane, let's say over here, is the watery environment on the outside of the cell. And on the other side of this phospholipid bilayer is the watery environment on the inside of the cell. So only the heads that are hydrophilic are going to be turning towards and interacting with the water. The tails are afraid. They don't like water, so they're going to be protected by the heads. If you had me for Bio 105, I compared this to a peanut butter sandwich. I love peanut butter. So, the tails are like the peanut butter, and the heads are like the bread. What better sandwich can you have? Peanut butter sandwich. If you take that peanut butter sandwich and you make it into a circle, right, that's your cell. One cell in your body. On the inside, that's the cytosol or the cytoplasm, and all the cytoplasmic organelles are in there with the nucleus and the mitochondria and the ER and all that. And on the outside, the watery environment of the extracellular fluid. So fats make up the membrane of each of the cells in your body.
So as things are trying to go into the cell,
Yeah.
what happens when they do eventually interact with the lipid?
Yeah, so there's certain substances that are going to easily be able to pass right through just the spaces within the phospholipids. There are certain proteins, and we'll talk about this in the next chapter in particular, that will embed in between the phospholipids and allow substances to pass in and out of the cell. There's hormones that are going to interact with receptors on the surface of the cell and then either be drawn in or elicit some change on the inside of the cell. It's all different mechanisms that can happen.
So if the lipids were actually polar, all the substances going through are going to maybe stay there and not make it to where they're supposed to go?
Well, yeah, I mean, there are certain substances, for example, that might just reach this head region instead. Sure. But depending on what the proteins are embedded in the membrane, they might pass all the way through. It kind of depends on what we're talking about. We can have a protein that just sits here on the inside of the cell membrane and interacts with those heads. Or we can have a protein that embeds right through. And remember how we talked about like dissolves in like? Right? So if we're looking at a substance that's hydrophilic, then it's more likely it's going to stay here and interact with the hydrophilic environment. If we're looking at something that's hydrophobic, like with cholesterol in particular, which cholesterol is a steroid, it's a lipid, just like the phospholipids are, hydrophobic, that cholesterol is going to embed itself here and interact with the tails, with the hydrophobic tails.
What happens with oxygen?
Oxygen and carbon dioxide in particular, or gases very small, they can diffuse easily right through the membrane. The polarity and all that, it doesn't matter. Water is another one. Water can move through the membrane, although most of the time there's a specific channel, an aquaporin, for the water to pass through. It's a special protein for the water to pass through. So how do we know if something gets into the cell? If you were making sugar cookies and you're using food coloring to color the sugar cookies red, and you get some of that food coloring on your skin, what happens to your skin? It's going to turn red. Why? Because what happened with the red color? It diffused through your phospholipid bilayer and got into your cells. If it doesn't, then we can assume that the membrane is impermeable to that substance. That substance, the red food coloring, cannot pass through the membrane because maybe it's too large, or maybe the charge is different, something like that. The conditions weren't right. But if we stain our skin with food coloring, whatever it might be, iodine, sometimes you'll see if you get an injection, they might rub iodine on your skin like an antiseptic, and it will stain your skin orange. That's the molecules moving in, passing through the phospholipid bilayer. We'll talk more about this in the next chapter or so in the process of diffusion and osmosis. You might remember that from bio. All right, so the actual phospholipid is a kind of different lipid because it has a hydrophilic part, which is the head, and then it has a hydrophobic part. But overall, if I ask you, are lipids polar or nonpolar, you should say nonpolar. Okay? Overall, they are nonpolar. The phospholipids have these two different regions to them. So here's the sterol that I was referring to before with cholesterol and the steroid hormones like estrogen and testosterone. Again, these are lipids. They're lipid-soluble hormones. They are nonpolar substances. They have a 4-carbon ring structure. anytime you see this 4-carbon ring structure, immediately you should think of a steroid, and immediately lipid. You do not need to recognize how the structure of testosterone is different from the structure of cholesterol. But you should remember that they are nonpolar and that they are lipids composed of a 4-carbon ring structure. So if I was going to administer something like testosterone or cortisol, and it's a lipid-soluble substance, do you think that that substance would have an easy time passing within the blood or a harder time? If you think about the common knowledge of blood, the majority of the liquid portion of blood is called the plasma. And the plasma is mostly water. So these steroids, these lipid-soluble substances, are going to have a more difficult time being transported in the bloodstream. And they will actually use transporter proteins, specific carriers, to help them move throughout the blood. Whereas if we were looking at a different hormone, I don't know, water-soluble hormone, that would have a much easier time being transported through the blood, but once it gets to the cell, it's going to have a harder time passing through that lipid bilayer. Cholesterol or testosterone or steroid hormone is going to have an easier time passing through the cell membrane because it is nonpolar, just like overall the cell membrane is nonpolar.
Is the hydrocarbon tail up there, is that like a fatty acid tail?
Yeah, it's similar, but it's not considered a fatty acid tail. But it is similar. It is carbon atoms with hydrogens to it.
So we're always going to see this in like a ring formation, whereas the lipid heads are going to be more like bonds.
Like circles, you mean, and lines?
Yeah, because this is rings of carbon.
Yeah, you will always see phospholipid like head and 2 tails. And the triglyceride,
Yeah.
flip back to it. I mean, the other picture is a little easier. Like, this is the head, and there's 3 tails to it. But the steroids you'll always see in those 4 rings. There isn't the head and tail here. There isn't that arrangement. You'll always see them in terms of the rings like this. There's no head and tail here.
We'll have to know how it like the difference between cholesterol versus cortisol.
You do not. You don't need to know, that to identify this as cortisol and this as testosterone. You don't. But if I showed you something like this structure, that you could tell me that that's a steroid as compared to a phospholipid or a triglyceride or even a carbohydrate at this point, you should recognize the groups. Just want to show you a picture from the next chapter because maybe it will make it a little bit easier for you to understand the proteins and how they're embedded within the membrane. Okay, so I just flipped to chapter 3. You don't need to worry about it. You didn't print the PowerPoint yet. Just to show you another view of the arrangement of the phospholipids within the cell membrane, in the plasma membrane. See the lighter blue circles? Those are the hydrophilic heads. These gray lines coming down, and some are bent and some are straight. Those are the hydrophobic tails. And then you can see the golden structure in yellow. You all see that? It's tiny, but you can see those 4 carbon rings. That's cholesterol. And because cholesterol is nonpolar, it's a lipid, it's going to interact with that more nonpolar environment of those fatty acid tails. And it's kind of going to get stuck in that region. And then all of these other substances, specifically the larger orange ones, are all proteins that are embedded within the membrane. Different parts of the protein might interact more with the hydrophilic areas, and parts of the protein will interact more with the hydrophobic areas. And each protein is going to have a job. So we'll talk more now about the protein structure themselves. Yeah.
Plasma in terms of lipids.
Say that again?
What was the significance of bringing up plasma in terms of lipids?
Because I want you to think ahead to when we talk about different structures like hormones and solutes and how they're going to move within the body, how we're going to maybe administer them in terms of medication, where we might give one as an injection versus one as an oral pill, and how it's going to be transported within the bloodstream. Is it going to just dissolve quite easily and move throughout the plasma, or are we going to need to transfer protein, and you'll see that now, to move that substance through the blood. So lipids can move through the blood with help at some point, yes. Lipid hormones, like steroid hormones, yes, will.
Okay.
But it all happens with help.
So, in terms of our body, if our diet is rich in saturated fat, and the lipid layer would then be the saturated, then say that's when cholesterol will have a hard time going through to the cell and then resulting in high levels of cholesterol.
Yes, and some of that's genetic as well. But yeah, definitely if your diet is rich in saturated fats, you're going to have a higher cholesterol reading overall. And you might aggregate those cholesterols and those fats into plaques within your blood vessels. And a plaque within a blood vessel is like a blockage. So rather than having this nice channel for blood to flow through, we have a block there. It makes less blood flow to an area, it makes it harder, and it increases blood pressure. So people with higher cholesterol generally have higher blood pressure, they're generally overweight. Yeah, we will a little bit, but mostly in A&P2. When we do reproduction and inheritance, that will be in the beginning portion of A&P2. Did you like that part? I did too. Yeah, I did too. A&P2, yes. A&P2, micro, 5105, principles of the
She
human
teaches
body.
micro only in the summer.
Sometimes in the summer, yep.
In the summer.
Not every summer, but and I really like it in the summer. I think it's a great course for the summer. Yeah, only focus on micro and nothing else for 5 weeks or so. Am I going to do it this summer? I don't know yet. I don't know yet. Professor Frost has been teaching micro here for a long time. So, you know, if she doesn't want to teach all the sections of micro, I might pick one up. Anyway, getting back to the 4 groups of organic. Okay, so any questions on lipids? And, you know, if you can't think of any right now, you can let me know after class or before the next class. All right, we'll look at proteins. So proteins are chains, polymers of amino acids. The basic building block of a protein is an amino acid. I entered amino acids probably the beginning of last class when we were talking about the functional groups. And the functional groups, the carboxyl functional group and the amino functional group in particular, you're going to see here with the proteins. Okay? Each amino acid has a carboxyl functional group and an amino functional group. And we're going to put these amino acids together through peptide bonds, which are special covalent bonds, to make up a protein. A protein's kind of like a necklace, where you have all different beads in your necklace, and they're all strung together. They're all bonded to each other. So we might have some beads that are red, and some beads that are green, and some beads that are blue, but they're all beads. Right? They're all going to be amino acids, but they're going to be a little bit different in terms of their structure. All right, so I'll show you some examples because I'm sure you're probably thinking of protein in the protein that you eat, in the meat or the chicken or the beans or the eggs, things like that. But for our purpose purposes, we're really gonna look at protein as a transport mechanism, as a structural piece of the body. We're not really gonna look at it as maybe an energy source like you might be thinking of it. Not really, because the lipid has a basic carbon, structure to it. Carbon, 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 36 carbons long in a fatty acid tail with all hydrogens. Proteins are different. They're gonna have a lot of different combinations of atoms bonded to them, not just hydrogen and carbon. For one, you already know that there's going to be oxygen and nitrogen there, but there's all different other combinations that you'll see. How branched chain amino acids are formed? Yeah, in amino acids, there's many different amino acids that have particular structures. You can have a branch off of the basic amino acid, and that would be a branched chain amino acid. I'll show you a few examples, so I think you're on the right track. So this is the basic structure of an amino acid. All right. Here's our carboxyl group. It's also called the Cu group. There was two forms of this that you had to know. It's the C double bond to the oxygen and single bond to another oxygen, and we could have a hydrogen here or not. Right? Two forms, they're both carboxyl. On the other side, here's the amino functional group. Two forms for this one as well. There can be the nitrogen with only two hydrogens, NH2, or there can be the nitrogen, as you see here, with the three hydrogens, NH3. Two different forms. Still the amino functional group. At one side carboxyl, at the opposite side amino. On one end of the carbon atom in the middle is a hydrogen atom that's always there. So it's these three that attach to this one carbon atom in the center. This is called the central carbon. And then there's an R. You saw that letter R before with the functional groups. The R is a variable group. It can be different. It can be different for every amino acid. Just like you had the functional group table and there was an R attached to the functional groups, it could be different. It varies. Okay. I don't know why they picked R rather than V for variable or I don't know. But it's R. It can be as simple as just a hydrogen atom right here. That's one amino acid. Carboxyl group, amino group, hydrogen, and another hydrogen here. That's one amino acid. It's called glycine. You don't need to know that. But there's another way. We can have a carbon here, and maybe three hydrogens attached to the carbon. So we could replace this R with a CH3. Right? That's an example of an R. Could be a CH3. That's another amino acid. It's not glycine anymore, because now I have CH3 here. Maybe it's alanine, different amino acid. I could have a, sulfur over here, or a carbon ring over here. I can have all different options that I can put. Yeah.
So the amino acid really requires carbohydrates to form. It needs that carboxyl group?
Not necessarily. So we're not really breaking down a carbohydrate to get this. We're using carbon, and we're using oxygen, and we're using hydrogen and nitrogen, but we're not making it from a carbohydrate. Yeah. Totally separate group on its own.
So those charges, they're not being applied to like the oxygen and the hydrogen, they're being applied to the group in general, right? Or not?
Both, really. So the negative charge on this oxygen is on the oxygen because of the way that it's functioning within this unit as a whole.
Okay.
Same thing with this positive charge here. And when we when I show you another amino acid bond to this one, you'll notice the charge is going to go away. So it's just the way that they're showing it to you right now, where you're just looking at one single amino acid. You're not looking at them bonded together. Once they're bonded together, this arrangement's going to change a little bit, and as well this one here.
Okay.
Yep. Okay, so this is just a diagram of some different amino acids, just so you can compare them. As I said, there are many different types, different kinds of amino acids. They all have this carboxyl group that's highlighted here in red or orange on the screen. See, they all have this carboxyl group. They all have the amino group that's in blue. They all have the hydrogen atom. You all see that? And then the R group, which they've colored in yellow, is the one that's different for each of these amino acids. This R group that's in yellow, this combination here bound to the central carbon that makes this amino acid lysine. And it's this R group here that's bound to this carbon that makes this one tyrosine. It's the R group that's different for each of the amino acids. You do not need to memorize the structures of these. When I was in school, I had to memorize, draw all of the 20 different amino acids in the structure. Can I do it today? No. No. Then, yeah, you better believe I did. But no, not anymore. But some of them, as you get to, have more experience with protein synthesis, you will just memorize just because you'll see them often. Glycine, like this one, you'll see this often. Okay. Alanine, serine, things like that, you'll just remember them.
The minimum carbon is 2 and the minimum is 90.
True. We could have 2 carbons if just these 2 existed, and the R group did not have any, just had a hydrogen atom. For the hydrogen, though, if we were looking at minimums, I would say the minimum would at least be 2, because you already have 1 hydrogen bound to the carbon, and then the most simplest R would be just a hydrogen. Even if there was a sulfur there or something else, there would still be at least a hydrogen there. So I would say minimum would at the very least be 2 to the central carbon, but you're still going to have 2 hydrogens to the nitrogen, and you're probably at some points going to have another hydrogen on the oxygen on the carboxyl. If I'm losing you, just ignore me for this moment. But does that make sense? Yeah. My question is never going to be the minimum number of hydrogen atoms in an amino acid. That's never going to be my question. But that's good. Okay, does this structure make sense to everyone? You can see how they're all different, how they're amino acids, how they look nothing like a lipid, right? And they look nothing like a carbohydrate, but they have carbon and hydrogen covalently bonded, so they're organic. So they're organic. You still have that carbon and a covalent bond to the hydrogen, no matter what one we look at. So let's look at putting them together. We want to make a protein. We already know the structure of the basic amino acid. You could see that with the glycine over here. In the glycine, you've got the central carbon atom, the amino group, the carboxyl group. Did I say hydrogen already? Hydrogen atom, and then the R. We already have this basic structure. But we want to take now that amino acid and peptide bond it to another one. Because a protein is composed of amino acids, not just one amino acid. One amino acid by itself is just an amino acid. It's not a protein. We want to have many amino acids to make a protein. If I have 2 amino acids bonded together, that's called a dipeptide. Similar to when we were looking at carbohydrates. And I said, well, we could put glucose and galactose together to make up sucrose, right? Or or whatever. We can make a disaccharide by putting two monosaccharides together. Two sugars bonded together is a disaccharide. Two amino acids bonded together is a dipeptide. Why peptide? Because the bond that forms in between the two is a peptide bond. So if we put glycine and alanine together, and you can see what they look like here once they're bonded, this is a dipeptide, two amino acids. If I add another amino acid to this, what do I make? Tripeptide. If I add another amino acid and another one, I might end up with a polypeptide. A polypeptide is a protein. For our purposes, it will really be 3 or more amino acids. In our body, typically you might see a protein that has 100 amino acids bonded together, or 50 amino acids bonded together. Is it collagen, I think, that has 51 amino acids. Hemoglobin has 146 amino acids. So each different protein has a specific chain of amino acids. Like I'm gonna make my necklace. I'm gonna start with my red bead. That might be my glycine. And then I might add yellow, and that might be alanine. And then I'm gonna add a green one, and that would be serine. And I keep going in whatever colors I wanna do to make this specific necklace. Because this necklace has a job. If I'm gonna make hemoglobin, I'm gonna start out with methionine, and then I might add, I don't know, glycine, and then alanine, and then serine, but I'm gonna have a particular order of these amino acids to make this one protein, because if I change the order of the amino acids, the protein's gonna change. It's like changing a recipe when you're cooking. You know, if you make a mistake and you're gonna make sugar cookies, and you make sugar cookies with salt instead of sugar, they're really not gonna be sugar cookies. Okay, yeah, they're gonna be salt cookies, right? So, you know, if you're gonna make something, you have to follow the recipe. And each protein is going to have a particular recipe to follow. If we change it, the protein is going to really not function the way that it should function. If it was hemoglobin, and instead of the 146 amino acids, maybe we added 140 different ones, it's not going to bind oxygen. It's not going to hold oxygen and transport it through the body. And how do we know what amino acids to add? How do we know to add methionine first and then add glycine? Right, our DNA tells us. DNA is what the instructions are. It's the recipe book. So to make all of these different proteins, collagen, hemoglobin, insulin, myosin, actin, all these different proteins, we're going to follow the instructions within the DNA. The DNA will tell us, for this protein you need this, then you add this, then you do this. That's what the DNA is for. DNA was going to drive protein synthesis. It's your recipe book. So the reactions in order to make this dipeptide that we were looking at before should be familiar to you. Dehydration synthesis. We talked about this term already. In the beginning of the chapter, when we were looking at anabolic reactions, animate, catabrate. And we also talked about it again when we did the carbohydrates. And we had the monosaccharide. We're putting those together to make up a disaccharide. Dehydration means removing water. Here it is over here. We're taking the two hydrogens and the oxygen away. We're dehydrating it. And synthesis, to make. So we're taking this water away. And here it is over here, our water byproduct, in order to put a bond in between the carbon atom from this carboxyl group, from glycine, and the nitrogen atom over here from the amino group in alanine. This carbon and this nitrogen are going to form a covalent bond with each other. This has all disappeared. The bond that forms here between the carbon and the nitrogen, it's colored in red. Doesn't really have to be colored in red, but they're just trying to highlight it here. That's the peptide bond. It's just a covalent bond, but it's between a carboxyl carbon, carbon from the carboxyl group, and an amino nitrogen, nitrogen from the amino group. Always a peptide bond, carboxyl carbon. Look for the double bonded oxygen. You see why I had you remember the functional groups? Right, carboxyl carbon and the amino nitrogen. That's a peptide bond.
And amino what?
Amino nitrogen. The nitrogen from the amino functional group. So here's another protein. Okay, we're making another protein here. And you can see in this representation, there's the carboxyl carbon, see, double bonded oxygen, and the peptide bond to the amino nitrogen. Here's the peptide bond. Here's another peptide bond. I've used arrows because sometimes it's a little bit difficult to see, especially if you're in the back and you're just looking at it. Now down here, kind of a darker green, these are the abbreviations for the amino acids that were used. So this is glycine, this is isoleucine, this is valine. You do not need to remember this. Glutamate, that's what these abbreviations are. It stands for the amino acid. Rather than show you the group, the R that's here, which can be many different combinations of atoms, they're just simplifying it and telling you, okay, well, this is the R group for glycine, that's here. This is the R group for isoleucine, that's here. Just trying to make it more simple for you.
So then that whole first thing would be glycine.
This part, yes, this whole first amino acid would be glycine. And if it was glycine, then this R group would just be a hydrogen atom. And then over here, like this one, can you guys see that? That's the amino acid for isoleucine. And over here, that's the amino acid for valine, and so on. And we just bonded them together. And at every point we made this peptide bond, we performed dehydration synthesis to do that.
And we lose hydrogen.
And we're losing water. Hydrogen and oxygen, water. We're losing water here when we made this bond. We're losing water here when we made this bond. We're losing water here when we made that bond. All right, so now we want to break this protein apart. Easier to think about it. You eat a protein and you digest the protein. You eat a chain of amino acids. We want to digest it. What reaction? Hydrolysis. So what do we have to add? Water. We have to add water back in so that we can then break these peptide bonds and digest the protein into amino acids again. So we have this long chain of amino acids. Glycine, isoleucine, valine, glutamate, and so on, this long chain of amino acids. And it has a particular order of the amino acids. The order of the amino acids, my red bead, then my blue, then my yellow, then my green, the order of those amino acids, that's called a protein's primary structure. Primary structure. Recipe, you put all the ingredients together. All right, you got your batter. What do you do next? You put it in the oven. And it changes shape, right? It grows. So if we're going to go from something that's flat like this to something that has a more three-dimensional, two-dimensional structure to it, then we need to change the way this is arranged. We need to form new bonds with these amino acids. And so far, we've only had covalent bonds here in this chain. When we start to add hydrogen bonds to the atoms within this beginning protein molecule, when we add the hydrogen bonds, then the shape's going to change. It's like you're putting the batter in the oven and it rises. Right? No longer are we going to have this flat chain of amino acids. It now might have areas of where it's folding or coiling. It's going to form more of a...
It's like a magnet between the different strands.
Yeah, it's kind of like, do you guys know what pipe cleaners are?
Yes.
Pipe cleaners,
Yes.
right? So you can leave the pipe cleaner flat, or you can twist it around your finger, or you could bend it, make little animals out of it and stuff, right? It's kind of like doing that. You know, we're taking something that can just lay there and be flat, and we're giving it a shape so that it can, overall, interact with something else. Maybe interact with the oxygen and carry it through the bloodstream, or interact with some hormone that's going to bind to it as it's on the plasma membrane of your cells, or interact with some other solute, allowing it to come in or out of the cell. We're going to give it a shape so it's going to do something, not just sit there. Somebody had a question? No? What else? Yeah.
Peptide bonds will be formed by carboxyl group and amino group.
Yeah, a peptide bond only between a carboxyl carbon and an amino nitrogen. It's a CN bond, carbon nitrogen bond. Yeah, only. So it's what I mean, right? Here we have the protein's primary structure. We have our flat necklace, just chain of amino acids. But then from this just covalent bonded chain of amino acids, we add in some hydrogen bonds in particular areas. And depending on where we're going to add those hydrogen bonds, you can see them here. Here's one hydrogen bond. Here's another hydrogen bond. The hydrogen bond is between this hydrogen here and this oxygen. Do you all see that? There's another one right here between this hydrogen and this oxygen. It's very small, but if you look closely, you can see that there are little dots. There's a dotted line to represent that hydrogen bond. Different from the little sticks, they're kind of like white sticks, that represent the covalent bonds. Through these hydrogen bonds, in particular the hydrogen and oxygen that are hydrogen bonded together, we can get a helix to form from that flat chain of amino acids. It can coil up. Or, hydrogen bond different ways, we can get a pleated sheet to form, as if you're going to take a piece of paper and fold it into a pleat, right? Give it a flat piece of paper, a three-dimensional structure. These are called the secondary structures of a protein. What was the primary structure? Just the amino acid chain. Okay. Secondary structure is when we can have an alpha helix or a pleated sheet. We're really not going to have both. Just those, yeah. Okay, so we might have from one protein and make an alpha helix. From another protein, we might make a pleated sheet. Tertiary structure. Knew that was coming. Tertiary means we're going to have a combination, basically, of those two. We're going to have some areas that might pleat and some areas that might coil. Some helices and some, pleated areas. A more three-dimensional structure to our protein. Notice they're not showing you the atoms anymore because it would be too complicated. They're simplifying it to just look like a ribbon. This ribbon is the chain of amino acids. And they're showing you the areas that will coil and the areas that will fold. Many proteins will stop right here. That's it. They'll go there, they're done. We baked the cake, we frosted it, it's done. You This protein might be fully functional at this point. But other proteins, we might need another step. The quaternary structure of a protein, hemoglobin, has a quaternary structure. It's like the fourth level. It's like making little roses out of fondant and putting them on the cake. It's like a whole nother level of decorating. So here in our quaternary structure, we have multiple polypeptide chains that are interacting with each other. So I not only made this cake, but I made another layer, and I'm gonna have them all interact, like the wedding cake down here. And each of these polypeptide chains has its own tertiary structure to it, and the way that they interact is specific for that protein. For hemoglobin, there's 4 polypeptide chains that interact with each other. And in the middle of each of them, we can have an oxygen.
So would that, for hemoglobin, would that be 4 separate proteins that are
Pretty
making
much.
one big protein?
Yeah. And we say 4 polypeptide chains rather than 4 proteins because the protein hemoglobin is really just the 4 of them together interacting.
So
Kara had a question.
So
Okay.
what would we have to know just to,
For questions?
just to identify which structure is which or what?
Yeah, you know, I wouldn't show you pictures of these, but I might say, is a protein functional at the secondary structural level? Not really, no. No, at the tertiary level it can. You know, or I might say, which of the following is an example of a quaternary structure? A dipeptide, a tripeptide, an alpha helix, multiple polypeptide chains, something like that.
So is it 3 or more and then 3 or less pretty much for the, so 3 or less polypeptide for tertiary and 3 or more for the quaternary?
No. Okay, so when I was saying 3 or more, it's really for a protein. There has to be more amino acids bonded together, at least 3 at the very minimum to make a protein. But for the tertiary structure, it's really just that one chain of amino acids that we were looking at, and parts of the chain are going to have helices and parts of the chain are going to have a pleated area. It's still only one ribbon. It's still only one chain, one polypeptide chain. But for quaternary, it's more than one polypeptide chain. It could be 2. I think they're showing you here 2 polypeptide chains. It's the blue one and the gray one. Do you all see that? They're both the same. It's just one's blue and one's gray, so that they're trying to make it a little easier for you to see that there's 2 of them interacting with each other. You made them both the same, starting from up here. 2 strands of pearls or two necklaces, and you twisted them together. So you have to at least have 2 polypeptide chains to interact to be quaternary. But it's quaternary because it's the 4th level of organization. Sorry if that's confusing. Yeah. Yes, Athena.
The protein isn't functional at the secondary, but it is a tertiary because of its ability to recognize and bind to someone alone.
Correct. Yep.
And the primary is not.
No.
So anything.
So do you see why bonding is so important? Right? If we didn't have hydrogen bonds, we would never get here. Right? You would never have proteins that function. Your DNA wouldn't be held together. So weak interactions, but very, very important interactions. Okay, so you think of proteins most of the time. You'll think of a protein as something that you eat, like meat or chicken or eggs or beans. If you look at an egg, right? If you were to crack an egg in a bowl, you've got the yolk of the egg and the white of the egg, right? But the white of the egg is really transparent. It's not really white until you cook it. The white of the egg is the protein called albumin. Right? Albumin is the protein in the white of the egg. So if I have albumin and it has a particular structure, chain of amino acids with maybe some folds and pleats and so on, and I cook the egg, I heat it up, then the transparent portion of the white of the egg will actually turn white, and the texture of the white of the egg will change. It gets harder, yes? Not liquid anymore, it gets hard. Taste is different too if you ever eaten raw eggs versus cooked eggs. So what you're doing essentially is you're denaturing the protein. And denaturing means getting rid of those hydrogen bonds. You're breaking the hydrogen bonds. We're getting rid of maybe the polypeptide interactions. We're getting rid of the alpha helix or the pleated sheet. We're going back to basics, back to the primary structure of the protein. When you cook an egg, what you're left with is the primary structure. When you denature a protein, you're left with the primary structure. You're not going to destroy that. Just by cooking it. So when you cook something, you're denaturing the proteins. You're changing the interactions. You're getting rid of the hydrogen bonds. When you flat iron your hair, right? That's not the reason I did this today, by the way, to make this point. I promise. You're getting rid of the hydrogen bonds. You're heating them up. You're denaturing the protein in order to get rid of that, to get back to that flat. So enzymes are proteins as well. Enzymes are another example of proteins. Enzymes speed up reactions. They're catalysts. They make reactions occur faster than what they would normally. You know, I could walk from here to the train station. I said could. I could. Okay, I could walk. It might take me 2 hours to walk there, but I'll get there. Or I could take the bus. Or I could drive my car. But I could get there faster, right? If I use some assistance, I could get there faster. The enzyme is that assistance. It's going to help a reaction to occur. It's not causing it, right? I'm going to go to the train station whether I got the bus or not. But it's going to help me get there faster. It's going to save my energy. It's going to lower the amount of energy that I need in order to get where I want to go. A reaction needs a certain amount of energy just to occur. If it uses an enzyme, it needs a lot less energy in order to occur. Usually the enzymes will have an ASE ending to them. So you'll see the name of something and at the end of it, it will be ASE. I have examples. Lipase, maltase, ASE. Not lipose, not maltose, right? Not glucose, but we're now putting an ASE at the end. If it was OSE, what would it be?
Sugar.
It's a sugar, it's carbohydrate. But ASE now tells you it's a protein and that's an enzyme. Enzymes are proteins. Pretty much all of the enzymes, at least that we will talk about, are proteins. Enzymes are proteins. But not every protein is an enzyme. Your hair is composed of protein. It's not really an enzyme. Insulin, protein, it's not really an enzyme. But enzymes are proteins. They speed up reactions. They lower the amount of energy required for a reaction to take place. Rob, you had a question?
Oh, I just crossed off most are proteins, you're still saying.
Yeah, I mean, the question, you know, enzymes are proteins, true or false? True. All proteins are enzymes, true or false? False. So looking at an enzyme, if I have an enzyme like maltase, right? What's in front of the ASE tells you what that enzyme's gonna do. So maltase is kind of like maltose, and maltose is a sugar, and maybe maltose is a disaccharide. So I might be taking these two monosaccharides and putting them together to make up maltose, to make up this disaccharide. And if I have this enzyme maltase, I could do that really fast and really easy with a lot less energy. Or lipase. You might be thinking of lipids, fats, putting fats together. I can either put them together quickly or I can break them down quickly. It works both ways. You know, forward of a reaction and the reverse of the reaction. I can make something fast or I can break it down fast. Same enzyme. By making maltose or I'm breaking it down, same enzyme, maltase. Forward and reverse of the reaction. I can use this enzyme repeatedly, over and over. It never gets used up. It's like an unlimited supply. Okay, I put these saccharides together, I made maltose, sugar. Now my enzyme's done, did its job, goes on. Oh, here's some more monosaccharide, I'm gonna put them together and make another one. And it can keep going. You can use it again and again. Yep, it's an unlimited supply. You know, all you can eat. This enzyme maltase has a specific structure to it. I probably drew this on the board maybe the first class or so. It has a particular structure. There is a chain of amino acids. It might be methionine and alanine and serine and so on that fold, that cleat, that coil, so that I have this chain of amino acids, and then they might fold, and they might coil, and they might fold again, and maybe I have another chain, and then they fold, right? So I have a tertiary structure now. I've got folds and I've got helices within that one protein. And in doing that, in making that tertiary structure, I've created these little pockets here. These two little areas where some things can bind to them or fit inside them nicely. This is called the active site. The active site of the enzyme. That's where the substrates are going to fit into. So if I'm looking at two monosaccharides and I want to put them together, then they're going to fit. One's going to fit here, and one's going to fit here. And the enzyme's just helping them to interact, helping them to come together.
Can you ever show us a picture of that?
I can. You know, you're not going to have to redraw this, because this is just an example. But you should be able to recognize it. I will show you another one. That's a protein, number one, because you can see the helices and you can see the pleats. And number two, that it has a tertiary structure, because you can see the both of them. And you can see the active sites, places at which something is going to bind to.
So if two monosaccharides bind with that to the enzyme, then do the monosaccharides bond to each other, and then the enzyme like goes
Says bye.
yeah, and then it's a disaccharide.
And then it's a disaccharide.
And now it's sitting there in the binding site?
Yep. It's in the active site.
And that, what reaction? is
Right?
the
So I
enzyme?
could have my A over here, and I could have my B, whatever it is, over here. And now they're kind of stuck near each other. Right? So then they can interact. We can bond them together. And now I have C. Remember the A plus B produces C? So I have my C I was not an art major, don't laugh at me. Okay? Right, I have my C, and that's gonna go off on its merry way. And what happens when it leaves? I still have the enzyme. Didn't change at all. And I can reuse it again. But this enzyme will only work with an A and a B. If I have something else, like D, it's not gonna work with that enzyme.
So this is the enzyme part, the blue part.
The blue part's the enzyme, yes.
Okay.
The A and the B are our substrates, and they can be monosaccharides, like I was saying before. These could be two monosaccharides coming together to make C make B maltose, right? Or they can be two other substances, two amino acids, or two lipids, or something coming together.
And then C could go back.
Yeah, and then we can also have C go back to this enzyme, bind to it again, split C, and we get A and B again. Is everyone with me? Do you all understand that?
Yeah, I have another question.
Yeah. I mean, my drawing is really not that beautiful, but so hopefully this will make a little more sense now.
Ms. Brown,
Yes.
we can use these enzymes again.
Yes.
Is there like a part of the body that's constantly making them, or is it just like, are they just there and
Yeah.
they just kind of
Both.
both?
Both. I mean, really, your cells are constantly making them, and it's driven just by DNA. When they're needed, we're making them.
Mhm.
When they're not needed, we don't make them. We don't waste the energy to do it. The amount of something is low. The amount of, I don't know, some protein is low within the body, will increase the production of it. Remember negative feedback? So if something is too high, we want to decrease it. If we make too much of something, we're not going to make more of it. But if we don't have enough of something, we're going to make more. So that we have a certain amount of whatever we need. Yeah. Somebody else had a question. No? Okay. All right, so if you look at the top, then you can see the two substrates here. Right. This is sucrose. This is just another example. I did maltose. Right? So sucrose, the disaccharide sucrose, it's bound to the active site of this enzyme. The enzyme is the orange structure. It's bound to the active site, and what happens? It splits it apart. It digests it into the two monosaccharides again. And what was added? Water at the top. So the hydrolysis reaction, when we're digesting a disaccharide or a protein or anything, we're always adding water in. This enzyme, whether we are using it in this direction to make sucrose, or we're using it in this direction to break sucrose down, it's the same enzyme that's working. Are we gonna make something or are we gonna break it down? It goes either way. This is what it actually looks like. And each of these little colored circles is an atom. So you might see white be hydrogen atoms, and the red carbon atoms, and the blue oxygen atoms, and so on. But you can see how they're all interarranged in such a way. Have you ever seen those little magnets? It's like a pile of little magnets, and you can like mold them into all different things and shapes and stuff.
Mm-hmm.
It's kind of like that, where they're molded in such a way that there's this little site over here, this binding site, that's the active site. All right, so questions on enzymes. They're catalysts, they speed up reactions, they lower the amount of energy required for a reaction to take place. They work for the forward and for the reverse. They're not used up. What else? They're specific for each reaction. I used my enzyme for maltose. Right? It's not going to work on lactose. We need a different enzyme for that. Specific binding site, specific active site on each enzyme. Okay, the last group of the organic compounds are the nucleic acids. The two that we will focus on are DNA and RNA. We will actually focus more on them a little bit later on as well. But for now, just basics. Okay, DNA, deoxyribonucleic acid. The Na stands for nucleic acid. RNA, ribonucleic acid. Deoxyribonucleic acid or ribonucleic acid. DNA and RNA, they're both in the nucleus. They're housed in the nucleus of your cells. DNA is kept there, kind of trapped there, but RNA, once it's made, is allowed to leave the nucleus. When we talk more about the cell and how it functions, we'll look at why that's true and what happens to it at that point. Both of them transmit, hold, pass on genetic information. The instructions that you need to make all the proteins, it's all found in the genetic information within the DNA and RNA. RNA, we say, is a transportable copy of the DNA. DNA you keep, it's your major instruction manual, major recipe book. But you might make photocopies of it and, you know, write some other recipes down and pass them out to your family. That's the RNA. Both of them are nucleic acids. They both have a carbohydrate. So they both have a sugar portion to them. They both have a phosphate portion to them, phosphate functional group. And they both have nitrogenous rings. It's like a carbon ring, but instead of carbon atoms, there's nitrogen atoms in the ring. I'll show you what I mean. So the very top, okay, you can see that square that's colored in blue. Here's a phosphate. In purple, that's our carbohydrate, that's our monosaccharide, our sugar. And then in yellow, this is the nitrogenous ring or the nitrogenous base. It's like the carbon ring but with nitrogens, and you can see the nitrogens. Do you all see that? Instead of carbon, there's nitrogen here, so we call this nitrogenous. It has a lot of nitrogen atoms. Both DNA and RNA have these basic structures, the phosphate group, the monosaccharide, sugar, and the nitrogenous rings. DNA and RNA. They're both built on nucleotides. We call them nucleic acids. They are built using nucleotides. Remember back to the protein. Protein was a polypeptide. It was built using what? Amino acids. Good. Starch was a polysaccharide. It was built using monosaccharides like glucose. Here we are looking at a nucleic acid. It's just like another chain, like another poly something, but it's a nucleic acid, and our building block this time is a nucleotide. Nucleotide has those basic parts: the phosphate group, the sugar, and the base. You might know already, DNA is in the form of a double helix. It looks like this. So we have these nucleotides that bond to each other, and they interact with each other through both covalent bonds and hydrogen bonds to form a double helix. Two chains of nucleotides that twist around each other, that link together through hydrogen bonds. This is one nucleotide. Right here is one nucleotide. Can you guys see that on your screen? That highlighted it? So one nucleotide, phosphate group, sugar, and base. Here's another nucleotide right here. Phosphate group, sugar, and base. And another nucleotide. Phosphate group, sugar, and base. But they're kind of flipped, right? On the other side. And another one. You have 4 nucleotides that you're looking at right here. The nucleotides are going to interact with each other kind of like up and down, as well as across. When they link up and down, they will link like this, like a zigzag. And we call that the sugar-phosphate backbone. So what's bonding together? The sugar and the phosphate, and the sugar and the phosphate, and the sugar and the phosphate. Those are all covalent bonds. Right? They're all linking covalently. That's what's forming this ribbon on the outside here. The purple part and the yellow part of the ribbon is the sugar and the phosphates. I don't know why they did not color it like that above. Being the OCD person that I am, I would have matched the colors. To me, I think, okay, the sugar's purple here, it's purple here. But the phosphate, in my opinion, should be yellow over here if it's yellow down here. You all with me? But at least you know what you're looking for, looking at right now. So the ribbon on either side with the purple and yellow lines to it, that's the sugar phosphate backbone. In the middle, we've got hydrogen bonds, the dotted line. Hydrogen bonds form through the interaction, I probably should change the color, but hydrogen bonds form through the interaction between the nitrogenous bases. It's not the sugars and phosphates anymore that we're looking at. Now we're looking at the nitrogenous bases, the rings with the nitrogen atoms in them. It's in between those rings that you'll see the hydrogen bonds. You see that? The hydrogen that's bonded to the oxygen, this hydrogen bond here, and the hydrogen that's bonded to the nitrogen, another hydrogen bond. That's what's holding the 2 chains of nucleotides together. So it's not just like the 2 necklaces that we were talking about that might be twisted around each other. They're not just twisted, they're actually hydrogen bonded together and twisted. Notice in this pair, where we are pairing this nitrogenous base that's red to the blue one, there's 3 hydrogen bonds. 1, 2, 3. You all see that? Right, I've highlighted in green the 3 hydrogen bonds. In the other nitrogenous pair here, there's 2 hydrogen bonds. There's one between the oxygen and hydrogen here at the top, and one between the hydrogen and the nitrogen here at the bottom. And we can have what we call base pairs within the DNA. This is what's holding the 2 strands together, holding the 2 chains of nucleotides together. I have 1 hydrogen bond to another. That's one of my base pairs, and in particular we call that the AT base pair. It's through the base adenine, see that here, and the base thymine. That's my AT base pair. It's held by 2 hydrogen bonds. And I can have another base pair. I can have the GC base pair. It's the guanine bound to the cytosine. GC base pair. The one between the blue and the red, which has 3 hydrogen bonds to it. So I can have these 2 options, these 2 base pairs. It's always the AT base pair and the GC base pair. And I could reverse it, and I could say the TA base pair or the CG. But I like the AT and the GC. I like it better in that order. It can can go either way. In my Bio 105 class, I said, I like to look at George Clooney. I think George Clooney is really cute. Yeah, I like to look at George Clooney. Does anyone know what I mean?
Yeah.
So ACT, I like to look at AT. That's your base pair. And George Clooney, just how perfectly does he fit there? GC. GC base pair. Alright. Yeah. But I do really like George Clooney though. I do. Alright, so AT base pair, GC base pair in DNA. Held through hydrogen bonds. Sugar phosphate backbone on the sides, the base pairs with the hydrogen bonds in the middle. And all of that's twisted into a double helix. Why do we say double? Why do we say double? Two strands, two chains of nucleotides. Good. Okay, and helix, obviously, because it's twisted. Questions so far on this? Okay, we'll talk more about DNA most likely when we get to the nucleus in the next class, and we will look at protein synthesis, the making of the protein, right? The making of a protein such as hemoglobin or insulin and how we're putting these amino acids together. We'll talk more about that process in the next chapter. About the structure of the nucleotide. I didn't talk about the pairing of trimine, but we will get to that. Look at the 4 nucleotides. ATP, your textbook puts here within this chapter, underneath nucleotides, or I should say after nucleotides, it is not a nucleotide. It is not a nucleic acid. It doesn't fit really into this description. So we'll talk more about ATP later, but it is not a nucleic acid. If that's not written on here, probably not. Please write that down. It's not a nucleic acid. This molecule, ATP, it does not carry genetics. It doesn't carry your genetic information. Only nucleic acids do. ATP is what we refer to as energy, right? Money in the bank, gold, paying for our reactions. And it looks similar to DNA or RNA because it has a phosphate, it has a sugar, and it has these nitrogenous rings. So it's similar, but it's not a nucleic acid. It actually has 3 phosphate groups, ATP, not just one. The sugar in ATP is called ribose. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. So the sugar is not the same. And the base, the nitrogenous ring, can only be adenine in ATP. In DNA and RNA, the base can be different. In DNA, the base can be adenine or thymine or George Clooney, right? G or C. In RNA, there's even another base. There's uracil in RNA. But in ATP, it's similar to DNA and RNA. It has a phosphate, it has a sugar, it has a base, but it's different in terms of its structure overall and its major function. Questions on this? Adenine is the base in ATP. The only one that there is. And we'll come back to DNA and RNA next class, and we'll talk more about some similarities and some differences. So we will look at it again. Take a 15-minute break. If there's no questions, come back, and we will do the lab on the microscope.